Morning. Welcome to Chem 103, module, what are we at now, 4? So today we're going to go back to this packet from last week. I want to go over some of the answers here and kind of explain how that works, going through some of the stuff that's been turned in. Again, I grade these based on your completion, not accuracy necessarily, but taking a look at them, I did notice some issues. So I want to spend a little time to go through this before the quiz next week. So first of all, just to kind of summarize the ideas here and what it is that we're looking at, we are trying to judge the forces between molecules. So these are not the bonds inside the molecules, but rather the attractive forces that allow molecules to sort of hold together in condensed states, that is liquids and solids. And one of the best ways we have to express that is the boiling point, because when you boil something, you separate the molecules from each other, but you don't damage or break the bonds inside the molecules themselves. So when we judge boiling point, we're judging these intermolecular forces, and so it's a good way to understand that and test it and so on. All right. So before that, before we get into that, let's just quick review the types of forces that we see here. And these are summarized actually after you make some of the conclusions here. Sorry, my thing is moving a little slowly. Okay, there we go. So there's the table. And remember we said that we can look at boiling point as a proxy for this idea of intermolecular forces. How strongly are the molecules holding on to each other? So let's again quickly take a look at the functional groups. We've got alkanes, alcohols, ethers, and we saw that for similar sized molecules, there's a big difference in boiling point. That alcohols have really, really high boiling points relative to their size. And then also we saw that in amines, that those have pretty high boiling points relative to their size. Ethers would have maybe slightly higher, but not much. And so we can also relate that to polarity when we look at the dipole moment. All right. So then going down here to the exercises, let's take a look at number 4. What does it look like to have a representation of a water molecule participating in a hydrogen bond with another water molecule? So here's our water molecule. And then we want to participate in a hydrogen bond. So in order to understand how that works, we have to think about what's going on with the polarity inside the water molecule. And what's happening there is that the electrons are pulled more toward the oxygen and away from the hydrogen. So we get a partial negative charge on the oxygen and a partial positive charge on the hydrogen. And this is what allows this molecule to interact with another hydrogen molecule because opposite charges are going to interact. So positive, negative, negative to positive. So our other water molecule might look like this, with that same type of separation of charge. And the other hydrogen would have a positive also. Right? So the electrons are pulled toward the oxygen in both cases, away from the hydrogen, which means the oxygen is going to be a little bit more negative and the hydrogen is going to be a little bit more positive. And because of that, we can show that there is this attractive interaction between the hydrogen from one molecule and the oxygen from the other. And that's what we call our hydrogen bond. All right, so wherever that is possible in a molecule, those compounds are going to have stronger forces, stronger attractive forces, and therefore higher boiling points. So it's going to take more energy to break apart those forces, so higher boiling points. So to kind of summarize all the stuff that you saw in this activity here, when judging boiling points, sorry, spell the letter there. When judging boiling points or intermolecular forces, we're going to look at two factors in order. First, we're going to look at types of intermolecular forces, because what we found in that activity is that molecules that can do hydrogen bonding are going to have the strongest forces. So that's going to be anything with OH or NH bonds. HF also fits in this category technically, but that's the only compound we ever see that in, so we don't really have to worry about it too much. So those are going to be your strongest. Then next, we have other types of polar forces. So anything that has oxygen or nitrogen or fluorine, chlorine, anything that's not just carbon and hydrogen is polar. And the examples that you saw in the document are the ethers and the ketones, which had stronger forces than the alkanes, but not nearly as strong as the hydrogen bonding. So let's just highlight this while we're looking at it here. So that's really what you want to look at first. When you've got hydrogen bonding in a molecule, you know that's going to be the strongest forces. So their other polar forces would be things like CO bonds, CN bonds, CF bonds, and there's lots of other things. Pretty much anything other than C and CH for our purposes, there are lots and lots of other things in the periodic table that would fit in this category, in that category, but for our purposes, that's what you're looking for. And then the weakest forces are going to be the ones that are totally nonpolar. So what we're going to see is molecules that are made primarily of CC and CH bonds without any oxygen or nitrogen. Those are going to be our nonpolar weakest forces. All right. So then once you've kind of ranked those, like let's look at an example of where that works and where we have to go on. So for B here, we're looking at three molecules, this one, and this one, and we're trying to find the highest boiling point, the strongest forces. So what we do is we look at those and we say, okay, what are the functional groups present here? What types of groups are there? There are CO double bond, that's called a ketone. There's OH, which is an alcohol, and there's NH2, which is an amine. So right away, we know that it's not going to be this first one, because we know that those general CO polar forces are not as strong as the OH and NH forces, which are the hydrogen bonding. So we know it's going to be one of these two that has the strongest interactions. And between those two, if we go back up and review, the OH is stronger than the NH. So the strongest here, the highest boiling point, is going to be that middle compound, ethanol, that's called ethanol, and that is going to have the highest boiling point and the strongest intermolecular forces. Okay, so where does that not work? Where does that technique not work? Well, let's look at C. The next one down, we see that every compound in this list, in this row of three, has an OH here. So that means that the comparison we just talked about isn't going to work because everything there has an OH. So instead, we need another way to rank them. And the second thing we're going to look at, if they have the same types of intermolecular forces, is the size of the molecule. Bigger molecules have stronger intermolecular forces. Now, that's a much simpler rule to understand and think about. Bigger means stronger. But what a common mistake is here, and one that I saw looking through some of your stuff, is that it has a second priority to the type of intermolecular forces. So you have to look at the types of forces first, and then if they're all the same, then you can move to looking at the size. You can't just look at size first, even though that's easier, because you'll miss the types of forces, which is actually a more important effect. It has a bigger effect than the size does. But when we have the same types of forces as we do in C, then we can look at the size and say, okay, well, this one's going to have the strongest forces because it's the biggest one. All right. So now let's look through the other three and take a look at those. So for A, it looks like it's going to be one that we're going to have to deal with size, but then we notice this third one actually has a second OH. And since these hydrogen bonding interactions, each type of each OH can participate in its own hydrogen bonding interaction. And so the more OHs you have, the more interactions you have, the stronger the interactions can be, and ultimately the higher the boiling point. So this one's going to be the highest boiling point here because of that extra OH, allowing it to do more hydrogen bonding. D, the three molecules are this one, and this one. And those are, see, now we know what this one is from the line structure stuff. And this is an example of where you need to just look at the types of forces. We see an NH right here. So this molecule can do hydrogen bonding. These cannot. That one's the strongest. Similarly in E, these are all the same size molecule, but the only one that can do hydrogen bonding is here with the OH. So that one's the strongest. All right, so hopefully that gives you a sense of how this stuff works. All right, and the next thing I want to look at is macronutrient structures. And to do that, I'm going to, if I can find it, I'm going to pull up my notes on this. All right, maybe that's broken at the moment. Okay, I don't know why these notes aren't coming up. But give me a second here, I'll have to restart it. So while that's going, the lab this week is about recognizing macronutrient structures. And so that is talking about the things in our diet that we need the most of. So specifically, we're talking about carbohydrates, lipids, and proteins. And so I wanted to talk a little bit about what those structures are and what they look like and how we can recognize them structurally. And if this doesn't come up, then we'll have to postpone this discussion, but okay, I think it's working now. All right, so I've got some structures of macronutrients. Let me pull them up. Here we go. I can also post these if you want to see them, or if you want this document online. So the three macronutrient categories, as I said, carbohydrates, lipids, and proteins. The other things that we need in our diet are known as micronutrients. So that would be things like vitamins and minerals, things that you just need very small amounts. But we mostly need carbohydrates, lipids, and proteins. Our macronutrients are the source of our energy and really what makes our bodies run. And so let's take a look structurally at what's going on here. So carbohydrates are called carbohydrates because they have the formula C and H2O. So you can always kind of break them up into carbon and water. So the oxygens and hydrogens are generally in that water ratio. And because back in the old days, the only way you could analyze anything chemically was to break it down into smaller things and then figure out what those smaller things were, they noticed that you could get this water coming out of them, and it was always in the same ratio, and so they were named carbohydrates. We can further classify those into monosaccharides, disaccharides, polysaccharides. The simplest sugars are monosaccharides, which are one single molecule, generally about this size. Glucose is a common one, but there are many other ones as well. They can have you can sometimes see them in a straight chain type structure or in this ring type structure. Sometimes the ring is drawn as I've drawn it here with the correct bond angles. Sometimes it's drawn more in a flat way like this. Which isn't structurally correct, but it is fine as like a diagram of showing which things are where. What I want you to be able to do is recognize the structure of, recognize that if you see a molecule that looks like this, you're looking at a monosaccharide, a simple sugar, a carbohydrate. You don't have to memorize specifically the structure of glucose or any of these other molecules, but you should know which categories the molecules fit in by looking at their structures. So by looking at the structure of glucose or any other saccharide, you can see this is sucrose, a disaccharide, that's table sugar, and it's made of glucose and fructose hooked together here. When these are metabolized or broken apart, then you have glucose and fructose separately. So some identifying factors here. Carbohydrates generally have lots of OHs. Pretty much every carbon is going to have an OH coming off of it. Not quite everyone, but just about. And so those can be helpful ways of identifying them. And then, of course, there are these ring structures. And the monosaccharides are going to be just one ring. The disaccharides will be two. And then these can further form polysaccharides where you have a whole bunch of these things linked together. And then those layers all connected with hydrogen bonds. And depending on the strength and the orientation, you can have things that we can break down in our body, and those are known as starches. So things like amylose that you get in rice or potatoes are these long polysaccharides that we then break down into simpler sugars in the metabolism. There are also polysaccharides like cellulose, this one shown here, where the interactions between the hydrogen bonds are so strong that our body actually doesn't break those apart. And so when we eat cellulose, we don't metabolize that. And that's cellulose is stuff like wood. The structure of plants are made from cellulose. Sometimes when you hear about fiber in your diet, that's really what's going on here. It's these big polysaccharides that are not digested so much as pass through your body, basically as they are. And that's why we don't eat wood or grass, things like that. You need a different type of digestive system, like cows, for instance, might have, that can metabolize some of those compounds. All right, so you got carbohydrates, monosaccharides, disaccharides, polysaccharides. Okay, the next class we're going to look at is lipids. Lipids are important to us in our class because this is where our biodiesel project comes in. Lipids are, vegetable oil that we started with, canola oil, in that experiment is a lipid. Lipids are fats and oils, but they're actually a much broader category than that. Lipids cover everything non-polar that in your body that is a nutrient. So it's not only fats and oils, it's also other types of molecules like steroids and cholesterol, other things that are non-polar. Here, for example, is what's happening to make what we call a triglyceride. So this big molecule in the bottom here is a triglyceride, and that's made of fatty acid molecules, which have a carbon double bond to oxygen with an OH on one end. That's known as a carboxylic acid functional group. And then this really long chain of carbon coming off that way, or coming off either way, but coming off next to that. And that's a fatty acid. So lots of carbons, usually anywhere from 12 to 20 some carbons in a fatty acid. If you take 3 of those fatty acids and you condense them with glycerol, this molecule here, then you form a triglyceride. And a triglyceride is what we call fat. Now, the canola oil that we used in the biodiesel synthesis is an example of a triglyceride. It's actually not just one triglyceride. As with many natural products, it's a mixture of different molecules, but they all kind of have this type of structure. They could have different length chains, double bonds, things like that, but they're going to have the same basic structure and the same backbone. So what we did was actually the reverse of this process. We took triglycerides and then we mixed it with sodium hydroxide and methanol, and the sodium hydroxide breaks this apart into the glycerol and the fatty acid, and the methanol actually caps this so that instead of OH, there's another carbon over here, and that makes it that fuel. So when we get more into discussing the chemistry of that project, then we'll see that again. We'll see a similar type thing. Fatty acids can be either saturated or unsaturated. I'm sure you've heard of saturated or unsaturated fats. And that refers to the number of hydrogens in the chain. So a saturated fat will have as many hydrogens as possible, which means that it does not have any double bonds in the chain, all single bonds, which kind of makes this nice straight zigzag, but overall straight chain. When a double bond gets put in the chain, it causes this turn or a kink in the chain, which changes the structure of the fatty acid. And so this, for example, is oleic acid, which is unsaturated and has this bend in the chain because of that double bond. Now, if we think about the intermolecular forces and the effect on that from these different structures, having this nice long straight chain allows those saturated fats to pack together very nicely and compactly. And that's why saturated fats tend to be solid at room temperature. They have higher melting points because of that good packing and stronger intermolecular forces. With unsaturated fats, you have a more difficulty in the fats kind of getting aligned with each other. And so the resulting intermolecular forces are weaker, and the melting point then is lower. So liquid fats like oils, like canola oil, olive oil, generally have more unsaturated fatty acids, which have those lower melting points. And then there was something that came up for a while, although we don't see it so much anymore, called trans fats. And what happens there is you can take an unsaturated fat like this, and you can partially, what's called partially hydrogenate it, and it causes it to kind of stretch out, but it maintains that double bond. And these fats were used in foods for a long time because, one, they resist going rancid, so they have a much longer shelf life. And two, there in the 80s, there was a big anti-saturated fat diet movement. And so people were avoiding saturated fat as being bad for you. And so you could sell trans fats as unsaturated, even though they pretty much behave the same as saturated. And now they know that these trans fats are actually much, much worse for you than saturated fats. And so they've pretty much been removed from the food system. Every once in a while, you'll still look at an ingredient list and it'll say partially hydrogenated something or other. And that's indication that there's some trans fats in there. So again, lipids are a pretty diverse category. And so I'd encourage you to look at the reading that's linked from Blackboard this week, where you can see some of all the different types of lipid molecules, the hormones and steroids and things like that also fit in this category. Another important thing about fatty acids is that what happens is they can make this interesting structure where when you take off the hydrogen on the left, you make this extremely polar piece known as a carboxylate. So you have a negatively charged oxygen atom here and another oxygen atom up here. So those are very polar and can interact with water, dissolve in water. But then it's still got this big tail, which is all carbon, 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 hydrogen. So that's nonpolar. And so these orient themselves into something called a micelle, where the head, which is represented as a sphere, is that polar carboxylate, and the tail, which is represented as this green line, is the long chain. And so then they make a ball, like a sphere, where all of those long chains point in, and the heads stay on the outside in a polar medium like water. And that allows these things to work like soaps. These are how soaps work. dirt and grease and stuff is mostly nonpolar. And so the micelles can trap that in the middle with all of their nonpolar tails. And then the outside can interact with water. And so each micelle can be dissolved in water. And that's why when you wash with soap, it's really the rubbing and the agitation that makes it work, because you have to break up all of these molecules and get all of the little dirt particles inside the micelles so then they can be washed away by the water. Okay, and then our final category of macronutrients that we're going to look at this week are proteins. Proteins are made of amino acids. All amino acids have this type of structure right here. So you have a carbon atom with something attached. The circle just means something. Sometimes that something is known as an R group. So sometimes we call that R, which just kind of means like wild card, like whatever. And then next to that, or bonded to that carbon, we have a carboxylic acid functional group. Bonded on the other side, we have an amine, which is a nitrogen compound, usually NH2, but doesn't have to be. And then so that's called an amino acid. And depending on what that R group is, that's what gives you all the different amino acids that you can possibly have. So for example, I'm going to pull up a picture here. Maybe might be too small. Let's see. Yeah, that'll work. So here are all the amino acids. And certainly you've heard of some of these: glycine, alanine, valine, leucine, serine, threonine, aspartic acid, tryptophan, tyrosine, phenylalanine, lots of different names. You don't need to memorize these or anything. In something like a biochemistry class, if you took something like that, or a physiology class, you might have to know some of these structures. But you can see here, let's kind of take a look at each one of them. So each one has a carbon in the middle. In this case, it's a line structure, so you don't see that. So that's that CH right there. And then the acid and the amine, and then the R group coming off. And in this particular table, the R group is coming off upwards. So glycine, the R group is just H. Alanine, the R group is CH3. Valine, it's this 3-carbon chain. Leucine, it's a 4-carbon chain. And so on. They're sometimes split up into these different categories. We don't have to worry about that. But these are all the amino acids. There's a subcategory of the amino acids you might have heard of called essential amino acids. Those are amino acids that our body does not make, and so we have to get them from our diet. It doesn't make them any more important than other ones. It just means that we need them from our diet. The non-essential ones we don't need. We make them from we just make them. Our bodies make them. Excuse me. So when these amino acids hook together, that's when we make proteins. And that first step is called a peptide. Back to our, coming back to our notes here. 2 amino acids can link together. The acid side and the amine side can link together into something called a peptide bond, also known as an amide. And that's what gives proteins their structure. So these chains of amino acids can hook together in whatever order. Since there's so many different amino acids, they can be lots of different possible combinations. When we talk about sequencing a protein, for example, we're talking about what amino acids are happening in which order. And then as those get bigger and bigger, most proteins are made from hundreds and hundreds and hundreds and hundreds of these things. They start to twist or arrange themselves in various ways with hydrogen bonding and form the big protein structures. And so if you look up the structure of an entire protein, grab a picture of that. you actually can't see the amino acids at all because you have to zoom out for this really big molecule. And so here's an example of a protein molecule. Might look something like that. And that doesn't look like anything we've ever seen in, we don't see any atoms or anything in this. But these twisty things they're called alpha helix, and they are made from these amino acids bonding in such a way that allows the hydrogens and the nitrogens and the oxygens to line up in hydrogen bonds, holding that twist together. And the same thing with these like ribbon looking things, which are called beta sheets. And those are also the different amino acids in those chains hooking together. And then the chain parts are just kind of less organized chain parts. And then the way this whole thing actually is organized also depends on the different interactions of the amino acids with each other. And so that's how you get this full protein structure, which just kind of looks like a random mess, but it's actually a very specific structure. And if this protein were to unfold, also known as denature, it would no longer do whatever it is that it's supposed to do inside your body. Proteins have lots of different functions. They can be structural, they can be enzymes, so they can facilitate chemical reactions, lots and lots of different things. They can fight disease. And if their structure is compromised, then they no longer do their thing. So one thing that you'll be doing that you either did yesterday in lab or you'll be doing next week in lab is to do some chemical tests on all of these different macronutrients to see if they're present in different food samples. And I think that lab was pretty successful yesterday for Group A, and so I'm excited to see how Group B does with it next week. All right, so that's a quick intro to macronutrients and reviewing some of the stuff from last week. Next week, we'll look at ions. Make sure you read some of those resources before that, because you probably won't. Well, I don't know that there's a specific activity you need to do this week. So I'll see you next week on Thursday. Yeah, and let me know if you have any questions or if anything else comes up. So thanks, everybody.